Before you start setting up your PCR reaction, it is essential to prepare a PCR protocol with all the details regarding the sample's order, the chemical ingredients, their volumes, and the PCR conditions. The samples are placed in the lower rack, whereas the ingredients are placed in the upper rack. The ingredients we will use in the PCR reactions are forward and reverse primer, water, dye, and a master mix. Then, before distributing the samples and mixing the ingredients, one has to vortex each tube and spin it down. Both the samples and the PCR ingredients are stored in the freezer. Therefore, one has to plan an extra time beforehand to allow them to defrost. To facilitate the distribution of the ingredients, we will prepare a PCR reaction mix in a separate tube. Please do not forget about labeling it properly. Afterwards, one should double-check the amount of each ingredient needed and transfer it accordingly to a PCR mix tube. It is usually a good practice to start pipetting with larger volumes. For PCR reaction, one should use nuclease-free water, which fills up the master mix to a desired PCR reaction volume. Then we add a master mix containing a reaction buffer optimized for the PCR reaction, DNTPs, free nucleotides used to build a complementary DNA strand, magnesium chloride, which is a cofactor for polymerase, and finally a Taq polymerase, which initiates the entire reaction. In this case, we use a hot start polymerase, which is only triggered by elevated temperature, making it easier to handle at room temperature. Then we add a coral dye, which stains the DNA and thus allowing us to distribute the PCR product directly onto agarose gel, where it can be visualized. Finally, we add a forward and reverse primer, in our case the universal BF3 and BR2 primers, commonly used for Marquezou benters. After all ingredients are added and before distributing them, one has to remember about thorough vortexing and centrifuging them down. The PCR reaction is usually performed in a special PCR stripe or a 69 well plate. To avoid possible contamination, one should not reach directly into the stripes package, but rather place them on a paper towel. Before distributing the PCR mix, it is better to close all the PCR straps with dedicated lids. It is also important to mind the orientation of both stripes and the lids, which should be consistent in a single run. It is crucial to label the PCR straps properly, preferably in more than one place. It is useful to include the date, the project name, and the number of particular PCR stripe. Now we are ready to distribute the PCR mix. We do it stripe by stripe. In this particular case, we don't have to exchange the pipette tips, since we are distributing always the same substance. Now when the PCR mix is distributed among the stripes, it is time to add the DNA template. First make sure that your DNA samples are in correct order. It is also very useful to order them exactly as they are in the preparation sheet. Apart from extraction replicates, which we have included during the DNA extraction, one should consider using also PCR replicates of each sample for better reliability. After all ingredients are placed in their respective well among the DNA samples, it is very important to close the stripes, vortex them, and spin them down before placing them in a thermocycler. Always remember about putting a proper counterbalance when centrifuging. Now we are finally ready to place our precious PCR straps in a thermocycler, where we set up the respective PCR program. Don't forget to put extra empty straps to have an equal pressure on the thermocycler's lid on all our samples. Most importantly, don't forget to fasten the lid properly. It's not over yet. Double check your PCR program and make sure it's running. After your PCR reaction is running, save time to put all ingredients back in the freezer in their respective places. It's also good practice to label open chemicals with a clear marking. The first step PCR is now ready. We unfasten the lid and take out the samples. Here we can check if there was no evaporation of the PCR product. Then we return to our working bench to vortex and spin down our first step product. Next, we distribute the PCR mix for the second step of the PCR. It primarily differs from the first step PCR mix with a lack of primers that we beforehand directly added to the mix. The second step primers are in fact sample specific and are added after the PCR mix to the specific sample. Again, don't forget about vortexing and centrifuging the PCR mix before its distribution. It is probably the most crucial step of the two-step PCR where one has to add the tagging primers. Tagging primers are selected combinations of a 6 base pair long known sequence which allows us to distinguish particular samples after the sequencing during bioinformatics analysis. To make things easier, our tagging primer aliquots are already containing both forward and reverse tagging primers. That is why we have to pipette them only once to a respective sample. The final step of the preparation of the second step PCR involves using this fantastic tool, which is a multi-channel pipette. With that, we are able to swiftly transfer first step product to the new stripes. To keep things clear, we maintain the original order of the samples from the previous PCR step. After the second step PCR is done, one should visualize the PCR product on the agarose gel by performing electrophoresis. Then, if all went well, the PCR product is ready to be normalized and be prepared for sequencing.